So what we're going to talk about right now is what's called the acute phase response, which is actually the systemic inflammatory response to infection. And again, this is part of our innate immune system. It's similar in many ways to inflammation, acute inflammation at the local level, but once an infection, if inflammation is being caused by the presence of a pathogen, once that infection gets to a certain size, it can cause a systemic inflammatory response. So if we start with an infection that occurs in some organ of the body, let's say in the lungs. Okay, so the infection is located in the lungs, and we have an acute inflammatory response at the local level to this infection, starting also at the lungs. Okay, so as this infection grows and grows, the inflammatory response begins to increase in size. And some of the inflammatory mediators, especially three key cytokines that are released, start to be released in greater and greater amounts, and they start to enter general circulation at concentrations that begin to affect other organs. So these three key cytokines involved in the acute phase response or systemic inflammation is interleukin 1, interleukin 6, and TNF alpha. So as these cytokines enter the bloodstream at concentrations that begin to rise and affect other organs, they start to bind to their receptors on some key target cells. Okay? And some of the key target organs that are affected by these cytokines and elicit the acute phase response includes the brain, specifically the hypothalamus. Okay. It also includes the bone marrow, and it includes the liver. Those are three key targets for systemic inflammation. So the effect that these cytokines have on the brain include things like producing fever, so elevating body temperature. It also produces what is sometimes referred to as sick behavior. And this is this general feeling of malaise, extreme fatigue, aches and pains. Okay? So all of the things that we feel when we are sick. Okay? So that is something that is caused by these cytokines and their actions in the brain. These cytokines also act on the bone marrow, and they induce the bone marrow to increase their production of scavenger cells, neutrophils, and monocytes. They act on the liver, and they increase the production of acute phase response proteins, which includes C-reactive protein and mannose-binding lectin, or CRP, and MBL. Okay. And those proteins enter the bloodstream, and they interact with complement proteins, and they increase the likelihood of complement activation. Okay. So those are some of the key components of the systemic inflammatory response triggered by these cytokines. So let's look at a little bit more detail of the actions of these cytokines. So these are the key target organs for these cytokines. As I mentioned, the liver, bone marrow endothelium, hypothalamus, and then there's two others that I didn't mention, but I will right now, which is fat and muscle and the dendritic cells in our body. So interleukin 1, interleukin 6, and TNF alpha, as I mentioned, they act on the liver and they increase the production of acute phase response proteins, namely C-reactive protein, mannose-binding lectin, and that helps activate the complement system. These proteins can also act as opsins. They can opsonize pathogens, increase the likelihood of phagocytosis by neutrophils and macrophages. Bone marrow responds to these three cytokines by increasing the production of monocytes and neutrophils, and that enhances phagocytosis and the actions of these scavenger cells. These three cytokines are also sometimes referred to as endogenous pyrogens, because they act on the hypothalamus, which regulates body temperature, and they trigger fever. And fever serves a purpose. Fever actually is designed to decrease the rate of viral and bacterial replication. Okay? So fever enhances the fight against these pathogens. IL-1, IL-6, and TNF-alpha also act on fat and muscle, and it causes the mobilization of energy. Okay? So it increases metabolism and it mobilizes energy, and this mobilization of energy and increased metabolism helps to support the fever, helps to increase body temperature. And then these cytokines also have an interesting effect on the dendritic cells in our body, particularly TNF-alpha. What TNF-alpha does is actually stimulate dendritic cells to migrate to the nearest lymph node. Okay, so whether or not a dendritic cell is triggered to, whether it phagocytizes a pathogen and is then triggered to migrate to the lymph node or not, TNF-alpha stimulates it to move to the lymph node, and that is on the off chance that the dendritic cell has encountered an antigen and has yet to trigger that response. Okay, so that promotes a more efficient and timely initiation of an adaptive immune response. So the acute phase response, or systemic inflammation, is really our innate immune system trying to fight this infection. Okay? It's trying to fight this infection by mobilizing all of our resources, okay, and enhancing the likelihood that the pathogen will be identified, will be presented to our adaptive immune system, and an adaptive immune response can be triggered. Okay. Unfortunately, this systemic inflammatory response can sometimes be quite exaggerated. Sometimes what can happen is that it can be triggered at such a high degree that the result is something that is actually very maladaptive and is something that we identify as sepsis. Okay? So this systemic inflammatory response at a scale that is that reflects tissue infection. Okay? So when an infection is in a tissue bed, a systemic inflammatory response is at a moderate level that is adaptive and helps fight the infection. However, if the, let's say this is a bacterial infection, if this bacterial infection leaves the tissue and enters the bloodstream, okay, so when the bacteria leave tissue and enter the bloodstream, and the bacteria and its components, like LPS, which is bacterial endotoxin, when the bacteria and its components enter the circulatory system, they are going to interact with key activators of inflammation, and they are going to trigger sepsis. And sepsis is essentially a greatly exaggerated systemic inflammatory response that ends up being very maladaptive and fatal. So the key cells that the bacteria and bacterial components are going to interact with in circulation includes the endothelial lining of blood vessels. Okay? The endothelial lining of blood vessels interact with the bacterial components and increase the release of tissue factor and platelet activating factor, and that's going to increase clotting, and that increased clotting can cause small blood vessels to be occluded by those clots. Okay? And when small blood vessels get occluded, then you have reduced tissue perfusion and tissue and organ death and failure. Okay? Secondly, these bacteria and bacterial components interact with circulating neutrophils. And circulating neutrophils release cytokines, especially TNF-alpha and interleukin-1. They also release reactive oxygen species, Okay? So you have these circulating neutrophils dumping reactive oxygen species into the vasculature. Okay? And then those reactive oxygen species are also promoting occlusion of the microvasculature because of damage to blood vessel walls. They are also creating instability in the vasculature, okay, because of damage to blood vessel walls. And when we say instability, it means that fluid is leaking out of cells. The cytokines released by the neutrophils, along with other mediators of inflammation, are also going to cause some of those vascular events of inflammation that we talked about, vasodilation, increased permeability. They're going to cause that, but they're going to cause it on a systemic level. So the vasodilation, increased permeability of inflammation is adaptive on a local level, but it is highly dangerous on a systemic level. Okay? The vasodilation, the increased permeability and leaking of plasma out of the blood vessels causes a dramatic drop in blood pressure, okay, and can end up causing shock. Specifically, in this context, we're talking about septic shock. Okay. The bacterial components are also going to interact with monocytes in circulation and tissue macrophages such that are in highly perfused organs like the liver. Okay? And they are going to have similar effects as we saw with the neutrophils. And then finally, the bacteria is also going to interact with circulating complement proteins. And it's going to cause significant activation of inflammation, which is going to potentiate the vascular events. So we're going to have even more vasodilation, even more increased permeability. Okay? And we're going to end up with a potential constellation of problems where we have leaking of fluid out of capillaries, significant vasodilation causing shock, or drop in blood pressure, which we understand to be shock, increased fever, sometimes extremely high fever, and problems with inappropriate coagulation. Okay? And so this is really the syndrome that we describe as sepsis, which can cause multiple organ failure, okay? Because all of our organs are receiving less blood flow because of a drop in pressure, because of inappropriate coagulation. And then and also one of the major complications of sepsis is a complication called DIC, or disseminated intravascular coagulation, which is extremely dangerous. And in DIC, it's actually a paradoxical hemorrhage, or paradoxical bleeding disorder. It's caused by an overactivation of the clotting cascade, okay, because all of these things are stimulating the clotting cascade. You overactivate the cascade, you run out of clotting factors because everything is activated. And then once you run out of clotting factors, you can no longer clot, and you end up with uncontrolled bleeding and potential hemorrhage. Okay, and that's DIC. So that's a serious complication of sepsis. So sepsis really needs to be interrupted at some point in the cascade, or else it's going to be quickly fatal. So the last topic in this chapter is to talk about the innate immune system's response to viral infection. While all of the things that we talked about are part of responding to a viral infection, there's also a specific group of cytokines and a particular group of cells that are important for responding to viral infections. And if you recall, I mentioned in last time's lecture, in our live lecture, that viral infections involve the virus injecting itself into a host cell and using that cell to replicate itself. So viruses occupy intracellular space. They're not going to be in the extracellular fluid. So one of the key parts of of triggering this response to viral infection actually comes from the host cell that is infected with the virus. So when a host cell detects double-stranded RNA, okay, within itself, that is a very clear sign to the host cell that it is infected with a virus. Now, double-stranded RNA, RNA that we produce, that our cells normally produce, is always single-stranded. Okay? And there's no normal occasion for our host cells to produce double-stranded RNA. However, many viruses are either enter the cell in the form of a double-stranded RNA, or part of the replication cycle almost always involves double-stranded RNA. So double-stranded RNA is a very, very strong cue of viral infection for our host cells. So when the host cell detects that, it releases two key cytokines, interferon alpha and interferon beta. And interferon alpha and beta are going to have effects on the host cell that released it, and also have effects on neighboring cells. So some of these effects include, so interferon alpha and beta, number one, it helps the host cell that produced the cytokines and neighboring host cells to produce enzymes that are going to degrade the viral RNA and block the production of viral protein. Okay? So this is going to help the host cell and neighboring host cells shut down viral replication. Secondly, the host cells are going to increase their production of a set of proteins that we call the major histocompatibility complex proteins, or MHC1s. And these proteins are important for the host cells to display the viral antigen at their membrane surface. Okay? And that displaying of a viral antigen at the membrane surface is going to be very important for the host cell to alert immune cells that it is infected with a virus. And then finally, interferon alpha and beta are also going to attract and activate natural killer cells. Okay? If you remember, these natural killer cells, also called natural killer lymphocytes, are one of the three major types of lymphocytes, and they are part of our innate immune system, and they are very important in fighting viral infections. So the natural killer cell is, once activated, is going to identify virus-infected host cells through this presentation of antigen by the host cell, and it's going to then kill those virus-infected cells. The natural killer cells also release interferon gamma, which is going to potentiate the effects of interferon alpha and beta. So all of the effects of interferon alpha and beta are made stronger by interferon gamma released by natural killer cells. Okay. Now, this activation of natural killer cells to help eliminate the virus-infected host cells is also aided by the fact that the macrophage released cytokines in the process of inflammation they also activate natural killer cells. So if this virus has already caused enough tissue damage to promote inflammation, these natural killer cells are already on the lookout for a virus-infected host cell. And so that's the key strategy for our innate immune system to fight virus-infected host cells. And if you keep in mind that these natural killer cells are fighting virus-infected host cells in a completely non-specific way. It doesn't matter what the identity of the virus is. It's going to just simply identify a host cell infected with a non-self antigen, and it's going to kill those cells.